Go. Hey, welcome to Modern Day Debates. Very embarrassing, and I'm very sorry. We just had a technical issue. I've tested this many times. You've seen we have done these streams using the software with the kind of the border image, background image. And so you've seen it done before successfully. I'm not sure what it is tonight, but we're just going to use Google Hangouts, and it might be for quite a few debates until we actually have everything taken care of with that new software. We're not going back until we know it will for sure work, because we're very sorry for taking your time. And we want to say thank you for those of you who have been so gracious with us. Last night, when we had to cancel that debate, I have to be honest, I felt terrible because it was like we were excited, and I was excited for the debaters, and I was excited there were people wanting to see the debate, and I was like, man, this is awesome. And then it was like, okay, this thing is not getting off. We're not getting off the ground here. And I just want to say thank you to you people who out there, friends who were supportive and saying even Natya Witness is an atheist viewer. And you guys know this is a Christian channel probably, or at least that I'm a Christian, and we try to be nonpartisan here. But I just want to say, you know, Natya Witness, an atheist, was kind of like, hey, how can I help out? And I was like, man, that means so much. Like, I totally appreciate that. And there were others as well in the chats that were offering help or being very gracious and forgiving, which means a lot. But it just, to me, it meant so much to me that it was like, regardless of worldview, it was like atheists who are showing grace and kindness. It was Christians who are showing grace and kindness and saying, hey, is there a way I can help? And that just makes it so much easier when I have those stressful situations like being on the road yesterday, trying to get it to work. And so I want to say thank you so much. This channel is like an abstract entity that belongs to everybody who's helped so much, because everybody has helped so much in terms of like people shoot me emails or like, hey, James, like you could actually do this, or hey, James, there's you know, someone somehow on the block list, you can pull them out if you go to this on the Creator Studio. Like, I honestly am all the time, other people, all the viewers are kind of steering the steering wheel, and I appreciate so much how everybody's just been so supportive. And like I said, we're going to do Google Hangouts for a while now until we can know that the new software will consistently work. We're not going to waste anybody's time. I have no idea what it was, but we're here, and we're thankful that you're here, and we're excited for this debate nonetheless, even though it's been crazy tonight. So I want to introduce the debaters, and then what we'll do is, once we introduce the debaters, I will talk about the format. We are debating creation evolution. And so whether you're a creationist, whether you believe in evolution, creation, whatever it is, we are stoked you're here. We want you to feel welcomed, and we want you to bring your ideas, and hopefully we get that diversity of views. So with that, I'm going to introduce the debaters. You already know Standing for Truth. He's been here before, and he's always ready to debate. He's ready to challenge people, and he's been reaching out to people, and we appreciate that. So Standing for Truth, I'm going to give you like 30 seconds just to talk about what's going on at your channel, and then I will give Adam 30 seconds to talk about what's going on at Steam Driven, his channel. And I'm going to put both of those in the new description box right now so that you can access those channels. And so without any further ado, glad to have you here, Standing for Truth, if you want to share what you've been up to.
James, thanks so much for having me on. I appreciate it, brother. Yeah, I think this is my fourth time on. I love your channel. I recommend it to everybody. I love how flexible you are and willing to put on these much needed debates. My channel, nothing too crazy. I'm having discussions as well. People seem to be enjoying them. They're a lot of fun. So I'd certainly recommend anyone go over there, subscribe. There's more to come. Lots of great videos, lots of great discussions in the future. I want to thank Steam, or Steam Engine here, for joining and filling in late notice. But yeah, overall, thanks so much, James. You're doing a great job with this channel, man.
That means a lot. Thank you for that encouragement during this stressful week. Totally appreciate it. And so also, Adam, this is your first time here, and we're really glad you're here, man. I mean, I'm glad that Standing for Truth was even talking you up. He was saying, hey, this is a well-read guy. He's smart, and he thought this is going to be just an awesome debate for everybody. So, Adam, thanks for coming on.
Thank you for having me. Thank you, Standing, for suggesting me. I wasn't expecting it at all. I got it about, what, 3-4 hours ago, saying, oh, come along, have a debate with us about creation evolution. I was like, well, fine, why not? Why not indeed? My background is, currently I'm in the forces. My channel is all about comedy, skits, gaming, nothing really to do with this sort of topic, but I'm still here and I will do my best for you. Thank you.
That's awesome. So we have an eclectic mix tonight, and that's awesome, man. I'm excited to see your channel myself. So we are going to get right into the debate. So I will read just the general format. So just so you know, we're going to have 10-minute introductions from each debater, then a 10-minute rebuttal from each debater, and then we're going to have a 10-minute cross-exam. So that will be just 10 minutes for each person where they're going to be drilling the other person with questions. That's my favorite part. And then we will have 5-minute closings followed by some question and answer. So that's going to be a great time. And so thanks for being here, everybody. Thanks for your support during this stressful time. Gosh, pulling my hair out. So thanks for your patience. And the Standing for Truth, we are going to give you the floor. So I'm setting the timer, and the floor is yours, Standing for Truth.
Awesome. Well, thanks so much, James. Can you see my my screen here?
You bet.
Perfect. Okay, so we got biblical creation versus atheistic evolution. You know what? The evolution that I imagine we're not going to agree on has to explain how that 4-dimensional genome with multiple overlapping codes and chock full of meta information came about. The term evolution, it can mean very different things. So let's define terms here. It can mean a change over time, but I think in regards to biological evolution, it means a change in allele frequency in populations over time or over generations. Now, the type of microevolutionary variation where you get a change in the frequency of the expression of different traits is not really disputed. This type of evolution explains the survival of the fittest, but not the arrival of the fittest. This type of evolution, if you're looking at my slide here, explains the small scale variation, but not the major innovations, the major origin events, or major new forms of life. Now, universal common ancestry, aka pawns come to people evolution, I believe is what we're going to disagree on in regards to the change. Now, I think it's important that you articulate your opponent's position correctly. So the tree, it's that great branching tree where the branches at the top of the tree represent the major life forms we see all around us today, while the side branches may represent various forms of life that went extinct a while back, and the root or the trunk of the tree would represent that one-celled organism at the very base of the tree, very far back in biological history, from which all the various biological forms of life are thought to have evolved or morphed or changed from that one simple form. Now, the question is, and the issue here is that cause of change. I would say mutations are destruction, not construction. And mutations in natural selection lack significant creative powers. All new innovations, structures, body plans, and novel information cannot be the result of natural selection acting upon random variation and random mutations. Now, they'll say given enough time, you'll get that population of non-dogs to turn into a population of dogs, but there's not enough time to see it. Time is no help for bacteria to biologist evolution. We're going down, not up. I find this picture funny because they'll say a frog can turn into a prince through lots of time, through lots of evolutionary processes. You know, that's science. But a frog turning into a prince instantly from the kiss of a princess, that's apparently a fairy tale. I would say they're both fairy tales. Now, a big thing here would be genome degradation or genetic entropy. If you think of it this way, we have a genome of 3 billion letters, and you take out one letter, change one letter randomly. Will you have a huge fitness effect? It's going to have a tiny fitness effect. And in fact, it's like rust on a car. You can't see each rust event, but it's continuous and destructive. We have genomic degeneration on a personal level and on a population level. We pass old and new mutations on to our kids and grandkids. It's 100 new mutations per person per generation, and nearly all non-neutral mutations are deleterious. We inherit a multitude of old mutations. Now, with this genome degradation, for numerous reasons, when it comes to rare beneficial mutations, they cannot sufficiently compensate for the relentless influx of deleterious mutations. No form of selection can stop genetic deterioration, or it can only slow it down through, say, natural selection. Now, problems not recognized by the neo-Darwinists is the limits of beneficial mutations. Natural selection is constrained by selection interference, and deleterious mutations are continuously pouring in faster than they can actually be removed. And based on what we know about the genome now, most mutations are not neutral, but are nearly neutral. Now, beneficials cannot keep up with deleterious mutations. And a lot of times you'll present examples, real-time examples of genome degradation, and they'll say, well, of course, that's inbreeding. You'd expect that. But inbreeding exposes the genetic mistakes within the genome, recessive mutations, right? Now, inbreeding, what it is it's like a sneak preview of where we are going genetically as a species. The reduced life expectancy of inbred children reflects the overall aging of the genome and reveals the hidden reservoir of genetic damage that has been accumulating. Now, the most important thing here is that genetic entropy is consistent with biblical creation. We're comparing models here. Genetic entropy is inconsistent with evolutionary theory, but consistent with the Bible, because the Bible records a time when people had extremely long lives. Now, when the lifespan of early biblical characters and their ages are actually compared, the biblical data clearly reveals an exponential decay curve. And this curve can only be described as biological. That means it must be concluded that the authors of Genesis either faithfully recorded an exponential decay of human lifespans, or they collaborated in fabricating the data using sophisticated mathematical modeling. And I find that hard to believe, because to fabricate this data would have required an advanced knowledge of mathematics, as well as a strong incentive to show exponential decay curve. So what is the most rational and logical conclusion given the empirical data? The data is real, and human life expectancy was once hundreds of years, but has progressively declined to current values. The most obvious and apparent explanation for such declining lifespans would be genetic degeneration due to mutation accumulation. Now, a lot of evolutionists will say, oh, genetic entropy has been refuted, but it has not. And when you look at rebuttals from evolutionists, whether it's Paulogia, whether it's a certain blog on the internet, Letters to Creationists, when fighting this truth, what their rebuttal will be, they'll speak of the obvious. Beneficials do happen. Selection does happen. Adaptation does happen. No one's disagreeing with this. Anybody with a basic understanding of genetics understands and knows these things. The argument of genetic entropy, genome degradation, only begins after acknowledging these obvious things. So let's look at some of the objections to genetic entropy. A lot of time they'll spend, and I know Jackson, we spent a lot of time saying that John Sanford misrepresented the data, misrepresented Camorra, but this is a rabbit trail. If you're looking at the slide here, John Sanford shows a larger zone of no selection than does Camorra, because Camorra's analysis of the problem was oversimplified. The crucial issue is about defining the correct distribution of mutation effects. The vast majority of beneficials will be unselectable. Anybody can easily see that most mutations fall within the no selection zone, and that almost all of them are deleterious. So even with strong selection, the entire zone can only undergo degeneration. Now, they'll appeal to beneficial mutations. That's what it comes down to. But due to the influx of deleterious mutations, the rare occurrence of beneficial mutations cannot compensate for the information loss. Beneficial mutations are very rare. Even evolutionists agree to this. And near neutral mutations are continuously and consistently deleterious, just as you would expect with typographical errors in a text. Most beneficial mutations are too subtle to be selected for by natural selection. Linkage group.
A minute and a half left.
Now, if you look at the examples, Thank you. Examples of beneficial mutations, if you're looking here, will not compensate for the information loss. It's about net gain versus net loss. Gene duplications are almost universally deleterious, and a few rare beneficial duplications cannot offset the many accumulating deleterious duplications, let alone all the other accumulating mutations. Evolution rescue devices, synergistic epistasis, mutation count mechanism, not biologically real, and simulations have disproven these rescue devices. Now, evidence for genomic degradation is everywhere. Lenski, he's revealing reductive evolution. His own data still reveals clear evidence of genetic degeneration. Homo naledi, Homo erectus, Homo neanderthalis, hobbits, all reductive evolution. Mammoth populations were highly inbred. There's a pandemic, entropic degeneration on all levels, Alzheimer's, dementia, autism, and so on. H1N1 went from a red hot pandemic to a whimper to an extinction event in 90 years. Linear accumulation of mutations is what caused this extinction. Super beneficial mutation to the rescue. Super beneficial mutations will not balance out the thousands of deleterious mutations. There's Haldane dilemma. Genomic data tells us that the human genome is profoundly different than apes. So when you actually calculate what would be needed for this type of evolution, it's impossible based on calculations. Genetics confirms biblical creation. We look at the three major haplogroups, all pointing us to Noah's three daughters-in-law. Created heterozygosity hypothesis. Was that it?
10 seconds left.
Created a heterozygosity hypothesis, gives us enough genetic variation and biodiversity that we need to explain all the life that we see on this planet. So that would be my presentation of the biblical model. And I think genetic degeneration is irrefutable. Thanks so much, James.
You are very welcome. Glad to have you as always. And so thank you for that opening statement. And now we are going to shift it to Adam, who's his first time here. And as mentioned, the debaters' links will be in the description so you can check them out. Highly encourage it. They've both been pleasant gentlemen, blokes, as Adam would say, maybe. So as I'm setting the timer, resetting it for Adam, just want to say if you enjoy debates, hit that subscribe button. We've got plenty more to come. And Adam, the floor is yours.
Thank you. I have had basically no time to prepare an actual 10-minute thing, so I'm going to improv. I've worked in a DNA laboratory quite a few years ago now, but I was studying cobra DNA. I was extracting cobra DNA from scales, blood samples, and it was to see the relationships between cobras from Asia and Africa. Now, I put them through all the tests, all the usual stuff. It took a hell of a long time because DNA extraction is an arduous process. But what I got out of it was a branching tree that showed all the different relatedness and the certainty of those how related they are in those final graphs. Took about 6 months to produce, so it is a long process. But that branch, or that series of branches showing these connectedness, is what we do for a huge portion of life. And that is one of the main reasons we let me try again. That is one of the evidences we have is the genetics, the makeup, the relatedness in terms of genetics. But not only is that good enough on its own, but it aligns up perfectly with morphology, archaeology, ecology, and basically everything. Every aspect of the biological sciences and even the chemical sciences line up with the things that we find in the DNA laboratories, things that I found in the DNA laboratories. I work a lot with reptiles, so if I come up with examples, most of the times it will be reptiles. Now we look at snakes, for example. Legless lizards is what they are. They belong to the larger lizard group, larger reptile group, even though you could argue that reptilia isn't a taxonomic group. That's something for another discussion. But if you look at ancient snakes, they had legs. And some snakes to this day, such as the boas and the pythons, still have things called pelvic spurs, which are tiny little claws just either side of the cloaca that have a very limited, if any, function at all. If you look at modern day skinks, they're going through the same process that the original snakes did all those millions of years ago, which they became burrowing animals. So they start to use their snout more to burrow under the ground. And if you see a blue-tongued skink, which is an Australian species, burrowing, it will tend to stop using its arms and legs, or just legs. It will put them to the side. And those legs have gradually shrunk over time. There are even skink species today that have just stumps where legs should be, completely useless, because they've adapted to this burrowing lifestyle. And we see this in all sorts of different species all over the planet, genera. And you could turn around and say, well, it's losing something. But put it this way. What do we have that the original cells not have? We're made up of chemistry, certain elements of chemistry, the same elements that would have been present in those first cells. There's nothing new about us chemically. Compare us to a fish. We have a head, we have bones, we have appendages. Look forward to something like Tiktaalik, which is one of my favorite examples. It has wrist bones. It has the bones that could allow it to move its head independent of its body. There is very little that you could say is different between us and them, apart from a massive shift in morphology, obviously. A few chemical changes here and there, hormonal changes, structures of bones. But it's all possible. There's not that much different. Creationists will always say, well, you need new information, you need these new structures. Not necessarily. The chemicals and the majority of the structures are already there from very, very early on. Speaking of Tiktaalik, I think one of the best examples of why evolution is the correct way to look at life is transitional forms. Tiktaalik being a good example of between fish and amphibians. It is a fish with traits of amphibians, but then again, we're also fish. We're Sarcopterygian fish. We're defined as that. So I think I want to argue that Tiktaalik is just a fish. Well, yeah, so are we. And look at us, we're completely different. One of my favorite examples also is Morganeucodon, which is a type of early mammal. If you look at the skulls of mammals and reptiles, they're different. The jaws, particularly. A reptile's jaw is formed up of several bones, but the mammal jaw is made up of just one. If you feel your jaw, that's just one bone there, but if you look at reptiles, multiple. Morganeucodon, as well as several other transitions, are interesting because you see the movement of those bones. If you took a reptile skull and a mammal skull and put Morganeucodon in the middle, you'd see three bones have shifted backwards almost to the ear, but they're still attached to the jaw. These are the stapes, malleus, incus. And you see a lot of these other transitionals where these bones are slowly shifting back through the skull until it becomes a truly mammal skull. So this is a mammal with a reptilian skull. Again, if you want to use reptilian as a taxonomic rank, which personally I don't believe it is, unless you count birds. Marsupials, I think, is a good example of evidence for evolution. The marsupials today are restricted to Australia, perhaps New Zealand, if I recall correctly. But they're not really found anywhere else in modern times across the globe. But if you look into the fossil record, if you look at the fossils and how they've been dated, you can actually see the migration of marsupials from Northern America down through South America, over Antarctica, into Australia. Back when the planet was, the tectonic plates were a completely different shift and you had different continents, these marsupials over time have moved down from North America through South America, over Antarctica and into Australia. And that's all corroborated by the dating that we used. So you see that as time has gone on, they've moved south, 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 east, 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 bang, Australia. And these are just a couple of examples that I can think of off the top of my head for good evidences for evolution. I'll address the genetic entropy stuff later, but I'm happy to concede the rest of my time here so we can move it along. Thank you.
You bet. So thank you. We really appreciate it, Adam, for that excellent opening statement. Very articulate for both of you. So now we will go into the next section, which is 10-minute rebuttal. So setting the clock. And one thing I'll let you know, Alexandra taught us this. It was a great idea. sorry, standing for truth, I cut in on you to say you had 3.5 minutes left. What I'll do in the future is I'm just going to, if you watch my picture, like my camera view, I will just let you know by the number of fingers that I hold up how many minutes you have left. Does that sound better?
Oh yeah, that works.
Okay, great. That way, no interruptions, and credit to Alexandra for her idea. And I am starting the rebuttal clock. So the floor is yours.
Awesome. Well, thanks so much there, Adam. You brought up some good points. This should be fun. I've got it all written down here. I'll start with Tiktaalik. Tiktaalik can't be, the transitional form between fish and amphibians because footprints of amphibians were found older than Tiktaalik. This is what the evolutionists are now saying about this apparent blunder. They're saying that we thought we've pinned down the origin of limbed tetrapods. We have to rethink the whole thing. That's surprising, but that's what the evidence tells us. He talked a lot about DNA chemistry. The thing is, we've moved beyond that, just like I said in my opening about the four-dimensional genome. It goes beyond chemistry. And it's pushing us to really conclude design. Like, for example, alternate splicing, for example, where individual exons perform different tasks to produce different proteins. We got protein moonlighting, like a multifunctional tool. The genome functions much like a computer. In regards to the relatedness he talked about, the initial prediction, though, of the evolutionary community before the Human Genome Project was that 80% of the DNA among all life should be conserved because of the millions of years. But instead, what we do find is that 50% is conserved. So massive failed prediction, which, forced evolutionists to develop this rescuing device, which is kind of like with the convergent evolution and a lot of the other rescue devices that they have developed in light of the new evidence. I mean, genetics, you can talk about genetics all day, but it's more and more confirming biblical creation. What we do know now in regards to, say, human chimpanzee genetic identity, which they used to say was 98 to 99% similar. The actual genetic identity now is only 88% similar, and that's 400 million DNA differences that exist between the two species. Another big one is human chromosome number 2, which they say arose through a fusion of two ape-like chromosomes. Now this purported fusion site is actually a functional DNA element in a human gene. They'll say humans and chimpanzees share genetic mistakes, aka pseudogenes, but now we know pseudogenes appear to be functional DNA elements, not mistakes. So genetics, if you want to talk the DNA language, the DNA chemistry, it's actually pointing to a biblical-based model. Also in regards to, say, the nested hierarchies that he talked about in the tree of life, the thing is, creationists too predict a nested hierarchy because it doesn't take much reflection to actually see that the young earth creation and evolution model make the same prediction. Because under the young earth creation model, God designed the entire universe, including the various kinds of biological life that exist in it. So obviously we'd expect to find that life fits a design pattern. And we know from the scriptures that we're made in God's image. So we can get a sense for what kinds of design patterns God might have used by examining the patterns that result from human design. So examples of these nested hierarchies abound among the design things in our world. That means that nested hierarchical patterns are as much the expectation of the young earth creation view as they are of the evolutionary view. So it's a competing explanation. Bones found in the dirt are not inherited, traits are. So when we look at this genetic damage based on this accumulation of near neutral mutations and the fact that there's no explanation for this net loss versus net gain, the fact is, whatever is being looked at in the geological record and the fossil record, it's based on a false assumption that molecules to man evolution is true, but based on what we know about genetics, it can't be true. And the ENCODE project itself shows that most nucleotides play a role in multiple overlapping codes. So if we're looking at beneficial mutations and we're comparing and looking at the net loss versus net gain, these beneficial mutations that are not deleterious would be vanishingly rare. And if you actually look at natural selection and beneficial mutations, we know that selection will remove the worst deleterious mutations and amplify the best beneficial mutations, but the accumulating damage is largely invisible, like rust on a car, like I said. So adaptations, they'll be highly visible. But Adam here, he's going to have to present us with, some examples of adaptation via, say, beneficial point mutation, duplication, whatever it is, or else he would fail to address the key issue, net gain versus net loss. So you can appeal to the fossil record, you can appeal to, these apparent transitional forms, but at the end of the day, if genetics points to a biblical model, then like I said before, it's just based on a false assumption. And if we look in here, if we look at even today, if we seen that finches, say, became extinct, and then some future paleontologists come across an island with fossil finches of, say, various beaks, and he has a pet theory that, say, early finches had short, stout beaks and gradually evolved to birds with long, thin beaks, he could then collect fossils that showed the early beaks and some with the later beaks, as well as fossils in between. So with that same logic as Darwinists have used on the, say, the fish to amphibian or reptile to mammal, as he talks about, or dino to bird even, with that same logic, he could actually make a case for his pet theory. But of course, he would be wrong in concluding that the long, thin beaks evolved in this hypothetical situation. So if we're looking at genetics, we need to see what the evidence actually points to. And the fact that our genome based on ENCODE is actually pointing to a genome that is fully functional, a lot of the evolutionists are actually against the evidence that ENCODE has discovered, because it contradicts their theory. And if a lot of what we're seeing here in the fossil record is being falsely interpreted, that means that this theory of molecules to man evolution can't be true, because the fossils are just circumstantial evidence requiring a theory that can actually account for how common descent could have occurred. So that means such a theory, as I've said in my opening, it must account for how the information in living organisms could have been built up in the process of common descent. Now, since the neo-Darwinian theory, nor any other theory presently known, is able to do this, we have to conclude that fossil evidence does not support universal common ancestry. Genetic entropy, deleterious mutation accumulation, among many more genetic reasons, make the fossil record nothing more than a circular argument. How much more time do I have, James? How much more, brother? Oh.
2 minutes and 45 seconds.
Perfect. Sorry about that. Also, many of the alleged transitional forms that he talks about do not have structures in transition from one form to another. Rather, the alleged transitional nature is a combination of fully formed structures that in themselves are not actually transitional. So Darwinian evolution would have to predict, it seems, a pattern where, let's use whale intermediates, for example, they'd have to appear progressively with sufficient time between them for new complex adaptations related to, say, an aquatic lifestyle to evolve. But this pattern is not what we actually see. We don't find the pattern that evolution predicts should actually be found in the fossil records, say, at certain times, but rather we find that truly aquatic whales, since we're using that as an example, appear abruptly. And even if we accept some of the fossils as intermediates between whale and land mammals, there is not enough time for the complex adaptations needed for whales' fully aquatic lifestyle to evolve. So whatever the correct explanation is, say, for the origin of a lot of these transitional looking creatures, unguided evolutionary mechanisms are not the answer, because natural selection acting upon random variation, random mutations can account for this, for all the life that we see. Also, in regards to, let me see here. You know what? I think I covered what I needed to from his thing. I can certainly end my time here. There. Thanks, James.
You got it. My pleasure. And with that, thank you very much for your rebuttal. So next we have the rebuttal, the first rebuttal, and this is from Adam, or Steam Driven. Stoked to have you give your rebuttal. So starting the clock, and the floor is yours.
Thank you, James, and thank you, Standing. I made some notes from your first bit, as well as a couple from the rebuttal you just gave me then, so I'll hope to run through all of that. The first thing I noticed, you put atheistic evolution on your first slide. There are Christians who accept evolution, so I think this a bit. I don't think you need to put atheistic in front of that. Just saying. So you said we don't know how we can explain how the genome came to be. This is very much an abiogenesis thing, because abiogenesis deals with the start of life, and the genome is essentially your sequence that replicates itself. So RNA or DNA. And it's commonly agreed now that RNA is probably where it all started off, RNA being a single strand of these nucleotides. And they have produced RNA in laboratories under certain conditions. So we know that it is possible for RNA to be created in certain environments. Usually, you've got to consider that there will be less oxygen in the initial environment, because a lot of the oxygen we have now came about when we first started to get photosynthetic organisms that produce a lot of the oxygen we have in our current atmosphere. That and a few other things, there are ways that RNA could have been created, after which you could have DNA within these phospholipid bilayers, which help protect them against water, which could damage them, and so on and so forth. I don't want to get too much into that. I've only got 10 minutes. Micro versus macro evolution. This is a very old one that I remember from my days of doing all this sort of stuff. So microevolution being change within species, and macro being change outside of the species level. We do actually see speciation. Ring species is a good example. I don't think anyone's ever managed to. This is a funny thing with dogs, Great Danes and Chihuahuas. Has anyone actually gotten them to breed on their own accord? Because if not, then I'm firmly of the belief that dogs are a ring species in themselves. Because they're only really species if there isn't some obstacle in the way. And I think the size difference between those two would be a considerable obstacle. So I like to consider dogs a ring species, and hence sort of an example of macroevolution being that it sort of crosses the species boundary. But there are many examples of speciation. Microevolution and macroevolution have exactly the same processes underlying them. So there is no fundamental difference between the two. Mutations, I put on him, mutations, destruction, not construction. I think what I meant when I noted that down was that you said that mutations don't construct, they don't add to a genome, rather they destroy it. This is a common mistake. If I say there are 10 green bottles sitting on my wall, and there was a mutation to one of them, and suddenly it was a blue bottle, then that's changed things entirely. I've not got 10 green bottles in my sentence. I've got 9 green bottles and 1 blue bottle are sitting on the wall. I've increased the information content there. There's more to it. What then if we have a duplication of the entire sequence of bottles after that? So I'll have the entire lot copied. I'll have 18 green bottles and 2 blue bottles sitting on the wall. And then after that, I could have 17 green bottles, 1 red bottle, and 2 blue bottles sitting on the wall. These are all things that we've observed in genetics, where you've got polyploidy, which is where chromosomes or gene sequences or entire genomes are duplicated and are then free to change. So when people say, oh, there's no method for increasing information, I'd say that 17 green bottles, 1 red bottle, and 2 blue bottles is more information than 10 green bottles. And I hope you'd agree with that. Genetic entropy is an idea come up with, thought by, I think his name was John Stanford, a creationist or an intelligent design advocate. I forget which one, but they're practically the same, I think. It's never been formally published. It's only been published in creationist documents as far as I can remember. And there's two problems with genetic entropy from what I know. It talks about how beneficial mutations can't keep up with deleterious mutations. But if you look at the process of how evolution works, that doesn't matter. If there are deleterious mutations in there that are so detrimental, then they'll be wiped out of the genome eventually. They'll be gone. If they're that bad, they'll disappear. Whereas the beneficial ones will maintain within the species or the genome, the gene pool. One other thing as well, they always use humans in genetic entropy, which is not by mistake. If you look around you in the world today, we've gotten to a point where natural selection, 4 minutes left, thank you. We've gotten to a point where we've sort of gone past natural selection. We have ways of saving people who in the wild or back 1,000 years ago wouldn't have a chance of survival. People with certain diseases, say Down syndromes or anything like that. We keep them alive, and a lot of them manage to reproduce. Now, they have genes that are not right, genes that have been badly mutated in some way, which has caused them to have this issue. What does that do to the average of the human genome? It will bring it down. That's why you get the declining lifespans. That's why you get the average fitness of the human race going down, because we keep the people alive, which is wholly a good thing. I don't disagree with it at all, but it does give you that false impression that somehow the human species is devolving. No, it's because our average is going down because more of us are surviving. That's how maths works. That's how you'd expect it to work. You said that Tiktaalik can't be transitional. This was in your rebuttal. You said Tiktaalik can't be transitional because amphibious footprints were found before it. That doesn't negate the fact that it's a transitional form. It's still something that is a fish but has amphibious traits. Surely it doesn't matter whether there's I haven't heard of these amphibious footprints before, so I'm just going on what you're saying. It doesn't matter. It's still an example of something that's a fish, something that's clearly a fish and does fishy things like fish do, but it has a neck and wrist bones. It's a transitional form. You didn't address Morganucodon as well, which is the example I pushed there. 2 minutes, thank you. Talk about you wanted me to give an example of a beneficial mutation or point mutation or what have you. I believe, correct me if I'm wrong. Well, later on. Look at the human skull compared to the skull of the other example, Tiktaalik. What do you notice is different? There are a lot of small bones in Tiktaalik's head, which means the head is more flexible. Look at our skull. It houses a big brain, and we live on land where there are a lot of hard things that can hurt us. Say, for example, in Tiktaalik, there was a point mutation, which meant that a couple of those bones in Tiktaalik's head were fused. That's going to increase the strength of the skull. It's going to decrease flexibility. So yeah, could be a problem. But as it's moving on to land, the fusing of skull bones can be extremely beneficial, because if a coconut bonked it on the head, it's not going to feel it quite as much, hopefully. So there's an example I can just think of on the top of my head. And lastly, before I finish, small against big beaks and finches, no. Because a lot of the fossils we do this sort of stuff on are dated. And we can see that one is a lot older than the other. If we did it with finches, we'd look at them and say, well, they're all from the same time, so we can't really make any judgments on that. Whereas something like Tiktaalik, fish, to land-dwelling amphibians, they all come with times attached to them. And you see Tulicpteran, Ichthyostega, Anthiostega, all these different transitions, all perfectly allotted in their time frame. And that's me done.
Excellent. Thank you very much. Very clear. And with that, we will go into the cross-examination, my favorite part. So we will let Standing for Truth start with the cross-examination. So we'll keep going with the normal order. And this will be 10 minutes. So this will be for each, and I am starting the clock, standing for truth. If you are ready, the floor is yours.
Okay, awesome there, Adam. Thanks for your rebuttal there. I guess my first question would be, because you talked about gene duplications and beneficial mutations, and I guess how many, if we were to look at numbers, how much gene duplications and say beneficial mutations are actually needed, per year for millions of years to account for the amount, the massive amount of living organisms that we actually see today. Because obviously just, claiming gene duplications can add the necessary novel information to the genome and can be beneficial enough to outweigh the genetic damage and take your fish to fishermen. Is there a paper that would detail, retrodictions on those numbers, or is it just more so a guess? Go ahead.
Well, you could look at the changing rates of DNA, and you can look at the divergence between, say, if you want an example of fish to humans, you could get your average fish genome compared to a human genome and say that's how different it is, and this is how we often, this is the rate at which we think these mutations occur.
Well, I think, keep going. Sorry, I didn't think you were done.
No, I'm done.
I think more so what I'm referring to would be like, even if we look at, say, Haldane's dilemma, like we know that a leading population geneticist, realize that even if there was an abundant and ongoing supply of, say, these beneficial mutations, natural selection must be very limited in its ability to amplify, right, such mutations to the point where they're actually fixed within a sizable population. So if you're going to, say, use gene duplication as your main mechanism for this large scale innovation, are there numbers, are there predictions, are there retrodictions, or is it just more of like a hypothetical or like an educated guess? Go ahead.
I think what you're asking me is, do the beneficial mutations account for us being here, despite? I'm not too sure, really.
Well, because if we look at genomic data, I'll try and make it a little more clear, right? Genomic data tells us that, well, we obviously know that the human genome is profoundly different than apes. So based on, let's say Haldane's dilemma, for example, there would need to be tens or even hundreds of millions of beneficial mutations that arose and then went to fixation during this time frame, right? So that scenario would require roughly, say, 1,000 independent beneficial fixations per generation. So according even to Haldane, that was almost an impossibility. So how is that resolved? Like, is there numbers? Is there any retrodictions? Because I guess that's what would make it scientific, right? Testable, falsifiable. Oh, can you hear me, Adam?
Yeah, I can hear you.
Like, do you find that Haldane's dilemma, for example, the problem of beneficial mutation fixation, I mean, is that resolved with actual scientific data, scientific papers, or in what you're saying in regards to, say, gene duplications, beneficial mutations, are there specific numbers? Are there papers out there that would break that down for us?
I imagine there could be. I invite you to go look for them.
Okay. If in regards to like building the genome, building that information that you're talking about earlier, I wasn't specifically referring to abiogenesis, but I did catch a few things on what you were saying in regards to DNA origins. From what I understand, evolution itself never actually predicted the RNA, DNA, protein system. And I think you said RNA evolved first, correct?
That's the common consensus, that RNA came before DNA, yeah.
So it sounded like you used a lot of, magical words like, probably, say, when explaining the RNA origins. And then obviously, if we're talking about testable, falsifiable science, that doesn't seem like it'd be science. Like, are there testable predictions and retrodictions on DNA origins? Selection pressure, for example. Are there actual numbers, or is this RNA, DNA, protein system, the evolution of DNA origins? Is this also more so like an educated guess?
The reason I spoke about it is because you brought it up in your rebuttal, so I felt the need to reply to it because you asked for it specifically. Aside from that, abiogenesis isn't my area, so my knowledge is to the best of it, and we're still not too certain on abiogenesis. So that's why I'm using deliberately vague language rather than saying this is fact.
And that's understandable too. I appreciated your responses there. Let's say with DNA redundancy, right? How did because we die without DNA redundancy. We know it's really important. How did evolution, know we die without DNA redundancy? Since evolution, natural selection looks at the short term, how did that evolve? What was the foresight in something like that?
I'm not too certain on that. What, DNA redundancy?
Yeah, just all the redundancy that we actually see in the genome. I mean, would you agree? Would you agree with the statement that overall now, based on new evidence, that the non-coding regions of DNA function, say, much like an operating system in a computer, right? It's directing timing, expression, regulation, and use of the other information or the data files that actually code for the protein. So, like, with natural selection's limited foresight, how did this computer-like code evolve? And how is that scientific? Like, if you go back to testable and falsifiable predictions, I mean, do you have any answers to that at all?
DNA is pure chemistry. Everything about DNA works within the bounds of chemical laws and everything we know about chemistry. So there's nothing about DNA that we wouldn't expect. Going back, it's all about the origins of DNA, because everything else is explained purely by the concept of chemical laws. So unless you're asking for the origins of it, I don't know what the problem is.
I would just say the building of a genome, say, from the single-celled organism. And I guess if you're talking about DNA chemistry, if we look at something like alternate splicing, which I think clearly shows design way beyond what evolution could do, how did each exon learn how to manufacture different proteins? Also, why are there bits of sequences teaching and regulating the exon? So it just seems like it's so much more than just chemistry. And I guess I'm just using the example of alternate splicing for now. Go ahead.
DNA is like an old football. It's a bit of a mess, really. There's scratches on it, scrapes. Bit of the fabric has worn off and disappeared. There's bits of grass stuck in it. DNA is a mess, and the only reason that can really be explained is by looking through the DNA of a whole host of different organisms and seeing how they compare, which bits are the same, which bits have changed, and comparing that to morphology, to the animals we have, how we see things today. That I don't understand what is you're asking, really, apart from that. How it's formed, how all these things came together. You're asking a very vague question of, I don't understand how this works. Tell me how.
I think I'm just saying that based on and in light of all the evidence about DNA function, it seems like we're dealing at this point with a highly compressed DNA code beyond the best computer code. We know we write books. We don't write books that can be read both forwards and backwards. So if, say, natural selection mutations had to build this genome, and I'm not specifically talking about abiogenesis, of course, but if evolution is true, given the millions or billions of years of evolution, I don't see how evolution could build such a highly compressed DNA code is what I'm saying. Like, do you find it more plausible to say there was no designer? I guess would be the best question in regards to that.
Put it this way. As I mentioned, there is nothing in DNA that doesn't follow the laws of chemistry. Therefore, we've got no reason to suppose it was something supernatural that doesn't follow the laws of chemistry that could have brought it about. We have to assume that because it all follows laws of chemistry, that it all follows laws of chemistry. And that's how it formed. We can't just say, oh, this is too complicated, therefore God did it. We can't do that. It's just not feasible.
But if DNA is a mess, as you say, how then does it produce, just say us, for example, so well? Like, for example, Hox genes?
Because when it doesn't produce as well, those organisms die.
Time. Sorry, I gave you a message in the internal Google chats, and then I also tried to hold up my finger. I hate to cut you off, standing for truth, but
That's okay. That's okay. Sorry, but we could probably go on with that forever. That was fun.
No problem. So, thanks for your patience. And then we will give Adam, or Steam Driven, his 10 minutes to cross-examine Standing for Truth and resetting the clock. The floor is yours.
Okay. Good starting point. Can you give me the different process in macro to micro evolution? What process is different?
So I would say if we're looking at micro and macro evolution, I don't personally like to use those words, because even in regards to our model of, say, created heterozygosity, right? God created, let's just use Adam and Eve, for example, with pre-existing DNA differences. So through processes like recombination, gene conversion, I mean, that would provide many new varieties quickly, since those differences are already built in. So I mean, certainly in regards to the biblical based model, I mean, we're going to predict change. Okay, and evolution means change over time, change in allele frequency in populations over time or generations. I think it just comes down to whether or not there's a limit to that change. So it's hard to say exactly what the difference between micro and macro is, because like I said in the opening, like that type of micro evolutionary variation where you get, a change in the frequency of the expression of different traits, I'm not disputing that, right? Because that does explain the survival of the fittest, but just not the arrival of the fittest. So I think it comes down to whether or not there's limits and how far you can take that change. Are dogs and bananas related or through common ancestry? I mean, I think there'd be a limit there.
Okay. So would you consider speciation to be part of micro or macro evolution?
I would say according to the biblical based model, speciation, say from shifts from heterozygosity to homozygosity, because that's how we would explain the rapid speciation since creation and since the flood. So speciation is important, and there's been documented cases recently. Actually, Dr. Nathaniel Jensen wrote a paper on it, where in real time, finches, it was observed that there were shifts from heterozygosity to homozygosity due to inbreeding, leading to a new species of finches. So that's actually what we would predict based on pre-existing heterozygosity, created heterozygosity, I should say. I'm not against that. And I wouldn't say that that's macroevolution, because I mean, there's still finches, there's still birds, but the change we've seen in that allele frequency in those populations of finches is expected and predicted under our model. So I think I wouldn't say that that's macro though.
Okay, next question. If we go back to my bottles on the wall example, would you consider that an increase in information?
Can you, I'm looking at my notes here. I have a lot of notes on everything you said.
Okay, yeah, I basically said you have 10 green bottles to start with. Then there's a mutation where one of those bottles becomes blue. Then you have another mutation where the entire sequence is copied. So you've got 18 green and 2 blue. And then there's another mutation where there's 1 red, 2 blue, and 17 greens. Is that an increase in information?
Well, it sounds like it's because what I was saying, shifts from heterozygosity to homozygosity was leading to speciation, say in a real-time example of these finches. So in your example with the bottles, I can't see what's going on a genomic level, on a DNA level, to see if it is a loss of information. Because if a lot of the examples I see of, say, speciation beneficial mutations, if it is decay of prior information, I don't believe that's going to take your fish to fishermen. So it's hard to say. I get your analogy with the bottles, but if you could reiterate the question a little bit differently, I mean, how can I conclude if it is an increase in information?
The analogy takes single point mutations, so the green bottle will change to blue, and polyploidy, which is the entire set duplicating, which we do witness. And it was just an analogy to say replace, I don't know, chromosomes with the bottles. And those are things we do see, we have seen. So I'm just asking whether that's an increase in information.
I would say that it's, let's say gene duplications, for example. I think, and we might be able to agree to this, Adam, I think gene duplications are almost universally deleterious. So say a few rare beneficial duplications, I don't see how those are going to be able to offset the many accumulating deleterious duplications, let alone all the other accumulating mutations. And with your bottle colors, those color changes in regards to our model, those are pre-programmed in our DNA. So that information is already present. There's no new novel and meaningful information added. And probably, like all examples of beneficial mutations, it probably is decay of prior information. Does that help answer your question?
I mean, I would disagree on it, but I'd like to ask another question.
Of course.
Got about 4 minutes left, and it's nice to have these little discussions, and they could lead on from there. But my next one was, how do you explain the gradual movement of marsupials from North America through South America, over Antarctica, and into Australia?
Okay, so actually, can you repeat that? So the gradual geographical distribution of marsupials?
We find fossil marsupials ranging from North America through South America, over Antarctica, and into Australia. And the way we date them shows that it happened over time. So it lines up perfectly, moving from North America through to Australia. Can you explain that?
Of course. Like I would say, obviously, if we're looking at, so you mentioned the fossil distribution of these marsupials. I mean, short answer would be, post-flood ice age, post-flood environmental conditions, because we look to the flood to explain the fossil record. So if we actually look at the upper layers, because you're talking about the upper layers where we're observing these marsupial distributions, correct? The fossils?
They do come from different layers.
Okay, so I would say that the layers that we see them in, based on our model of post-flood ice age, we see in those layers post-flood diversification and dispersion, and I think it's readily explainable through. I'm not sure if you've read much, creation literature, technical papers, but we say that it's explainable through land bridges, floating mats during the ice ages, right? And we can see that animals can be distributed around in very natural means, even in real time, say during cataclysmic events. So given a flood model and flood geology, I don't see a problem in that geographical distribution. Go ahead.
So you say that marsupials went from the Middle East, they traveled to North America, then down through South America, then over Antarctica, and then to Australia within... Well, because we know that no records of marsupials being in North America or Antarctica have been found by humans in recent times at all. So say, give it 1,000 years, max. Is that possible?
So 1,000 years since the actual post-flood ice
since
age.
The flood.
And you're trying to think, and you're saying that the distribution of these marsupials is
it's
conflicting
linear.
to... Yeah. And based on the remodeling of, say, the Earth post-flood, well, for one, I know you mentioned, dating methods. I mean, I'm sure that could go on a rabbit trail, so we won't go on that. But I would say that there's a lot of assumptions and guesses in there. So, I mean, for me, I disagree that would be reliable. But yeah, ice age producing land bridges to cross and the distribution that you see. But the thing is, these animals, thousands of years ago, based on our model of pre-existing heterozygosity, I mean, the variation, the biodiversity, I think would be even in excess as compared to what we see now. So yeah, are you saying that there's a lot of variety in these marsupials in these geographical related areas?
Yeah, they're not the same as today's marsupials.
Yeah, and I wouldn't expect them either, because shifts from hetero to homozygosity for us in explaining speciation, I mean, post-flood land bridges, post-flood environmental conditions, because you can't look at the Earth now and assume that that's, you know, exactly the way it was before. So I think that is a plausible explanation for the geographical distribution of not only marsupials, but all other life and diversity that we see as well.
Okay, one more question to finish us off.
Yeah, of course.
What's the point of a skink with stumps? I mentioned earlier a skink that has merely a couple of stumps near the front of its body that have no purpose whatsoever. What's the point of it?
I'm sorry. So it's a couple of stumps in front of its body that has no purpose?
Stumps. They've lost the claws. There's just a stump on either side.
Well, I know you mentioned, because you know, I would say a loss of information. You know, the evolutionists will use a lot of examples of losses of information as if, hey, lose everything and we'll gain it all. But in regards to that exact reason as to, I mean, if it's a specific mutation that made them lose the information, and it's a deformity, is what you're saying, correct?
It's a mutation. It's not a deformity. No, it's an actual species of skink. I forget its exact name, but it's a species that has stumps.
I mean, maybe we don't know the purpose yet. I mean, for example, how many vestigial organs were, you know, listed even 100 years ago? I think there was 140. Now they're all known to have a function. Now we're seeing this in the DNA language as well. So do we know, and do we have conclusive evidence that it has no purpose?
Time.
They're pretty useless. We'll finish there.
Excellent. So thank you both very much. And now we'll move into the quick closing. So this is just 5 minutes each. And to keep it moving, we will jump right into it. So Standing for Truth, if you have your materials ready, the floor is yours.
Awesome. Thank you. I think this has been fun. I really enjoyed the cross-examination. I didn't really see any real solid rebuttal to say genome degradation. I think that's important to explain the net loss versus the net gain, because if on a genetic level, for example, Darwin, he didn't know anything about genetics. You know, the laws of inheritance certainly came from Mendel and his pea plant experiments. Look into that. That certainly explains and reveals the evidence for our created heterozygosity model. But if there's no explanation for the genome degradation, obviously Pons Come to People evolution can't be true. So even phylogenetic systematics, it's based on a primary assumption that, you know, this universal common ancestry did happen, when we know based on genetics that it couldn't have happened. Now, that means, you know, a biblical based model based on the fact that genome degradation, mutation accumulation after the fall is consistent. The evidence, the empirical data is better explained through our model. And even if we look at, you know, examples of beneficial mutations, I know he gave an example of a skull, but I think even his example of the bottles there, for example, we have pre-programmed a lot of these changes. They're non-random epigenetic changes based on the pre-existing information. So that can't be used as sufficient examples for Pons Come to People evolution. And even if you look at the praised examples of beneficial mutations, whether it's, you know, African resistance to malaria, we know that's due to a broken hemoglobin protein. Melano mutation is praised as a human mutation, but we know that's a loss of net specificity. Nylon digestion by bacteria, also a loss of substrate specificity. And at the end of the day, if you're using examples of, say, adaptive immunity, homologous recombination, antibiotic resistance, and bacterial sickle cell anemia, as I said, you know, these all fail to meet the requirements of a gain in information. And if you're duplicating an already degenerate gene, you know, that's just going to speed up the degeneration process. And I didn't see him, you know, address the evidence that I provided for genomic degeneration. I mean, we see it in real time. You know, I know Richard Dawkins praises the E. coli experiment as strong evidence for evolution. I mean, we know Lenski demonstrated, and he is demonstrating reductive evolution. All beneficial mutations seen in this experiment, the reductive, loss of function, loss of promoter, loss of genes. They're painting themselves into a corner. They're handicapped, they're lazy, they're weak. They're getting rid of genes short term, but what it is it's long-term degeneration. So at the end of the day, the adaptation in this experiment, and this is a perfect experiment to look at to see whose model is stronger, it's largely accomplished by means of loss of function and loss of regulation mutation. So that's a net loss of information. That's not going to take your fish to fisherman. We see paleogenomic evidence. I mean, mammoth populations, we see that they carried an elevated genetic load. Neanderthal was highly inbred and had very high genetic load. There's a pandemic of mutation-related diseases in the human population: Alzheimer's, dementia, autism, asthma, autoimmune cancers, immunological. This is entropic degeneration on all levels. Without an explanation on how to solve this, you're not going to get your fish to fisherman evolution, and that means it's just a false assumption based on really a lack of evidence. And even the H1N1 human version, people will say that, you know, John Sanford misrepresented the data, but evolutionists themselves cannot explain why the H1N1 virus is extinct in humans. Right? Adaptive evolution does not cause extinctions. And the virus started off more human in its codon usage and ended up randomized. It became worse at interacting with human DNA over time. So if it is using the wrong codons, that means it will be less efficient at replicating in human hosts. So this is a decline in functionality and matches exactly with genetic entropy. So the amount of evidence for genome-wide degradation for John Sanford's model is actually overwhelming. And more importantly, genetics confirms biblical creation. If you look at the three major haplogroups, the length of the lines on the distribution of these mitochondrial DNA variants really does show there's three independent lines of evidence that fit exactly what we see in mitochondrial DNA. That's exactly what we'd expect through biblical creation. The history of humanity is in our genome. So what we know about mitochondrial DNA of humans fits the 6,000-year timescale, explains the three major haplogroups and the relative differences between pre and post-flood humanity. So I see a ton of evidence for biblical creation. The evidence that I see for biblical creation contradicts evolutionary theory. So at the end of the day, I think that's all I have to say. I think the model's strong, and I think the model for large-scale evolution is quite weak in light of the genetic data that we have. Thank you.
Thank you very much for that closing statement and resetting the clock. We'll try to keep moving fast. As one of the debaters we found out has to get up super early, we're going to try to move through this as fast as possible, and then we're going to try to do some questions and answers. It's going to be really short, though, to be honest. So we're going to try to get through as many questions as we can. Thanks for your patience on that. And the floor is yours, Adam. Thanks so much. Steam driven. Go ahead.
Thank you. Yeah, I've got to be up for work in 4 hours, so I won't be around too much longer. Evolution is chemistry. I mean, people will say it's biology, but everything about evolution is based entirely in chemistry. The interactions between various molecules in the DNA and outside of the DNA in the cells that make us, everything follows those laws of chemistry. And nobody's ever come up to me with an example of a mystery that chemistry cannot solve when it comes to evolution. We are answering the questions such as abiogenesis. That's the next big stepping point for us, I think, even though it's not technically part of evolution. I think it's going to be a big reveal when we finally get to it, when we finally say, this is how it's likely to have happened. And it will be chemistry that answers that question. Nothing that was said today has been much of a problem for me. There are things, obviously, I don't know, but I know that I can go look up. Evolution is an incredibly large subject. Incredibly large, and you'd be extremely naive to think that anybody would know every single part of it. But the people who doubt evolution, I suggest go listen to this debate and hear the points, and go look up the papers on it. Go look up John Sanford's paper. It's not an actual paper, as I've mentioned. Genetic entropy has never really been formally put forward. But go read it anyway. See what you think. And then look at what the scientists say. Apart from that, thank you very much.
Thank you very much. Totally appreciate it. Steam driven. So, as mentioned, both the debaters' links are in the description, even if you don't agree with them. Let's say you're like, man, I can't stand the other side. Well, this is a great chance to at least understand their perspective by going to read their own sources, and you can say truly stepping in their shoes as much as possible to get their point of view. So with that, we will start going into the question and answers. and we'll try to get through as many as possible while trying to respect our debaters' time. So if that's okay with the debaters, if we just go through like a short little bit, is that all right?
Yeah, we could just do a few questions. I understand he has to be up early, and we've gone for quite a while. So yeah, but whatever works best, brother.
I'm perfectly fine with it.
Okay. So we actually have a number of them. So Landon Freeman actually posted this before the debate started. So it is relevant though, and so I'll ask it because it's like pertinent to the material. But Landon Freeman, thanks for being proactive, checking out the cards, the event cards. So what evidence, he asked, this is for Steam Driven, he said, what evidence would cause you to doubt the naturalistic view of evolution and universal common ancestry?
It's quite a difficult question because there's so many areas that I could come up with. I could use the same example that Bill Nye used. I think it was Bill Nye, or might have been Richard Dawkins, of finding a fossil rabbit in the Cambrian. We could find that, well, yeah, essentially anything like that. Find any animal, any fossil that does not fit with what we have. Because everything we have found does fit. That's basically it. If something doesn't fit, it don't fit.
Gotcha. You bet. Well, thank you for that. And HistoryNet is asking, this is one for Standing for Truth, coming after you. This is an in-house objection, you might say. And HistoryNet also asked me, he asked, why haven't you had any Christians who believe in evolution come on to debate? And we're trying to do that. I'm actually reaching out to a scholar right now, not literally right now, in this 24-hour period, to ask if they'd be willing to come on and debate. So it'd be Christian versus maybe be an atheist, maybe be another Christian. So just so you know that, HistoryNet, thanks for that question. Now HistoryNet has a question for you, Standing for Truth, which is, why do you persist with denying evolution when most believers accept it?
Well, I think I'm not sure if most believers do accept it, because the believers that I've known, especially on a personal level, and in the church I go to, I mean, nobody accepts Ponscombe to people evolution. There's absolutely no evidence for it. I mean, I've debated plenty of scientists. Jackson Weed had a month to prepare. R.J. Downer, all great debates, great guys, great discussions. But literally, I mean, I've read all the rebuttals for genetic entropy, and 90% of the stronger rebuttals say, you know, mutation count, mechanism, synergistic epistasis. A lot of the examples that you think someone who's well read and prepared for a debate would actually use haven't even been used, and all they speak of is the obvious, right? You know, natural selection happens, adaptation happens, beneficial mutations happens. We're not arguing that, because the genomic degradation problem, genetic entropy, happens after the fact. So if I'm looking at the empirical data and see clearly that Ponscombe to people evolution can't be true, and the evolutionists that I assume are strong apologetics and the evolutionary heavyweights of YouTube also don't have a rebuttal to it and aren't even using the recommended rebuttals, why would I reject a perfectly good Bible? That's what I don't understand about theistic evolutionists is they reject a perfectly good Bible, reject what Genesis clearly teaches about man being created in the image of God, but yet they want to believe that man and apes are related through common ancestry over a dumb theory that has absolutely no evidence for it. So I reject it based on the fact that, for one, the Bible doesn't teach monkey to man evolution, and the empirical scientific data refutes bacteria to biologist evolution. So that's why I reject it.
Thank you for your answer, and thank you for your question, HistoryNet. We appreciate it. And so we're just going to ask one more, and then let's see. Just to respect the debaters' time, the debaters are really the ones who get in the hot seat and do so much work. And so I want to say thanks to both the debaters, and I also want to say especially thanks for Adam, for you're already kind of staying past time, and so totally appreciate that. And this is just the last question. So Pine Creek asks, if Standing Truth were not a Christian, does he think he'd be more likely to accept evolution via common descent?
That's actually a really good question. I know there are a lot of Christians who don't accept universal common ancestry. based on the empirical data. I mean, a lot of advocates and proponents of intelligent design aren't necessarily Christians. And when I started looking at the evidence, I was not, I didn't consider myself a born again Christian. I had not put my faith and trust in the work of Jesus Christ alone. So I wouldn't have even said I was saved. And from my research, I was already beginning to reject universal common ancestry. So obviously the Bible and Christianity does have something to do with it, but with the evidence that I've seen and the research that I've put into this, I don't see how it can be true for so many reasons. So no, I don't think, if I had the same research that I did now and I wasn't a Christian, whether I was a Muslim or a polytheist, whatever, I think I would have rejected it as well. It's a good hypothetical. That's probably the best answer I could give.
You got it. Excellent. So with that, I want to say thank you, everybody, for coming by. Thank you for your questions, which I will save. So in other words, once we have these debaters back, hopefully in the future, maybe in a month from now for a rematch, these questions are going to be saved. So it's not like I'm erasing them. I'm sorry we didn't get to all of them. And we thank you for them. We thank you for the debaters so much for getting in the hot seat and the preparation you've done. Just thanks for coming on. Glad to have you.
It's been an absolute pleasure, both of you. James and Standing, it's been wonderful talking to you, and I stayed up late, but I'm happy I did. So, and I wasn't prepared, so I'm still happy I did. So,
yeah, I enjoyed the talk. I think you did great. Maybe we can do it again in the future. Prepare a little more, but yeah, James, as always, you put on a good show, brother.
Thanks, I totally appreciate it. And if this is your first time here, highly encourage you hit that subscribe button and that little bell notification button for reminders of when we go live, as we will have debates almost every night this week. So it's going to be a pretty busy week, and we're stoked about all the different topics. So with that, I want to say thanks everybody for coming. And after show. So Standing for Truth is going to host an after show on his channel that I'll be headed over to. And then also Steve McCrae is also hosting an after show. So let's see, we will encourage you to check out both. Actually, you can have both windows open and you can kind of peek in and see what each is saying, as I'm not sure, but my guess is Steve McCrae might disagree. So Standing for Truth will probably disagree with Steve McCrae. So encourage you to check both of those out, and we hope you have a great night. Thanks for stopping by, and thanks for being so helpful to the channel, seriously, all the time. We're kind of learning together, and I'm ultimately learning because of all of you. So thanks for your help, and have a great night. Keep sifting out the reasonable from the unreasonable.